A very good morning to everyone. So today we will now continue the next lecture of pharmaceutical analysis. That is the next topic of unit number 2 is non-aqueous titrations. So far we have studied about aqueous titrations. All those titrations till now which we have studied were taking place in aqueous medium. Previously we have discussed about acid-based titrations. Acid-based titrations, as we have seen that we used acid to neutralize a base, or we used a base to neutralize an acid. This is all we have studied in the previous lectures. Also we have studied a very well-known equation, and we have studied different indicators which are used for performing acid-based titrations, and the theories related to the as indicators, that were Oswald's theory and benzoic theory. Now we will discuss non-aqueous titrations. Why there was a need for non-aqueous titrations? Basically, non-aqueous titrations are those which take place in other solvents except water. Water will not be used as the medium here. Water will not be used as a medium here. Other solvents other than water will be used here as the medium. So in non-aqueous titrations, we perform titrations which do not contain water. So why was there a need to go for non-aqueous titrations? The reason behind this is water molecule contains H positive ion and OH negative ion. Water dissociates into H positive ions and OH negative ions. These H positive ions and OH negative ions can compete with other weak acids and bases, and hence it may result in competition, and hence some error may take place. So there was a need for non-aqueous titrations because the H positive ions and OH negative ions resulting from the dissociation of water interact with other weak acid or base, and hence they compete in the proton acceptance or proton donation with other weak acids and bases. Moreover, we have certain compounds which are non-polar in nature. Certain organic compounds are completely non-polar in nature, and hence they cannot be dissolved in water. Their solubility in water is very, very poor, so we cannot dissolve them in water. So hence there is a need to go for substitute of water, and hence non-aqueous titrations are used for the estimation of those compounds which are itself water insoluble, and hence they will dissolve in other non-aqueous solvents. So we will start now with the non-aqueous titrations. So the need for non-aqueous titration arises because water can behave as a weak base also and a weak acid also, because water can dissociate into H positive ions also and OH negative ions. So it can show a dual character. It can behave as weak base also, and it can behave as weak acid also, and hence it can compete in the proton acceptance or the proton donation with other weak acids and bases dissolved in it. So this was the major problem in the aqueous titrations. When we use water as the medium, the proton ions and the hydroxyl group ions can compete. They can interact with other drug which is weak acid or weak base, and hence it can compete in the proton acceptance or proton donation, which may result in the error. Now, the procedure for non-aqueous titration is very useful because it satisfies two different requirements. Non-aqueous titrations are very useful because it satisfies two different requirements. The first one is suitable titration of every weak acid or bases, along with providing a solvent with the ability to dissolve organic compounds. So a non-aqueous titration, it solves our two purposes. The first purpose is that we can easily perform the titration of those compounds which are weak acids or bases. Along with that, non-aqueous solvent will also act as an organic solvent in it. So these are the two important requirements which are satisfied by using non-aqueous titrations. because of with the use of non-aqueous solvents, the interactions of the proton with other weak acids and weak base ligands will decrease, and hence there are lesser chances of error. Secondly, the non-aqueous solvent which we are using will act as solvent, and hence it has an ability to dissolve many organic compounds in it, since organic compounds, because of the different nature, are insoluble in water. So, I will illustrate the problem which arises when we use water as a source of medium, when we use water as a solvent. This is an example in which we are using water, which is not acting as a suitable solvent because for example, I have taken here amine group. I have taken a compound containing amine group, and as we all know that in amine group the nitrogen contains lone pair of electrons, so it will attract the positive part of the solvent towards itself. So when we dissolve amine group, this here I have taken a secondary amine. The secondary amine will it has tendency to interact with the proton which has been released by the water, and the amine group will be protonated. The protonation of amine group will take place, and we can see that because of the water, the reactant has changed its nature. Okay, now this reaction was not supposed to take place. This was not the required reaction which we wanted to perform, but because of the H positive ions which were dissociated by the water, this undesirable reaction have taken place. So this is a problem in many other reactions also, when we have when these the protons or the hydroxyl groups which are released by the water, they interact with other compounds and result into undesirable reactions which are not required. So this is the reaction which is competed with an aqueous solvent by the reaction given by H. So what happens is we sometimes this reaction which has taken place is competing with the other reaction which was supposed to take place. Now half of the concentration of H positive ions, half of the H positive ions have interacted with amine group, which was not our required reaction, which was not a required reaction, and half of the H positive ions have been merely used up in the reaction of amine group, which was not required. So this undesirable reaction is competing with the main reaction which was supposed to take place, that is the formation of hydronium ion. So hence we can say that this other competing reaction is going to result in error, and hence the error must be removed, and the error is removed by using non-aqueous titrations. In the non-aqueous titrations will be those in which we will not be using water, and we will be using other solvent other than water, and hence all the errors which were supposed to take place when we are using water as a medium will be eliminated. Now, non-aqueous titrations. This type of competition provided by water towards weak bases and weak acids makes it difficult to detect the endpoint of the titration. As we have seen, undesirable reactions take place, which makes it very difficult for us to detect the endpoint of the titration. Most of the drugs which we consume act as weak acid or a weak base, and hence the competition this type of competition, as we have studied previously in the case of amine group, happens for other drugs also which contain lone pair of electrons, and hence it results in undesirable reaction. Therefore, these substances which have very sharp endpoints when titrated in aqueous solutions due to their weakly basic or weakly acidic nature generally need to be titrated in non-aqueous solvents. No doubt there are many solutions which produce very sharp endpoint in aqueous medium as well, but still we substitute it with non-aqueous titrations because of only those competing reactions which are taking place, which are undesirable, and hence they need to be eliminated. Next is many reactions which occur in non-aqueous titration procedures can be explained via Bronsted-Lowry theory. So the non-aqueous titrations can be explained by Bronsted-Lowry theory. According to Bronsted-Lowry theory, acid is any substance which is capable of giving H positive ions, and according to Bronsted-Lowry theory, base is any substance which can accept proton. So this is as according to Bronsted-Lowry theory, acid is a proton donor and base is a proton acceptor. So non-aqueous titrations are based on this theory and its definitions of acids, and so this is our non-aqueous titrations are based on the Bronsted-Lowry theory and its definition of acids and bases. Basically, acids can be thought of as proton donors, whereas bases can be thought of as proton acceptors. So, according to this Bronsted-Lowry concept, which substance will be acting as proton donor? It can be HCl, it can be sulfuric acid, it can be nitric acid. All those species which can release H positive ions will be acting as Bronsted-Lowry acid, and the Bronsted-Lowry bases will be all those compounds which can accept proton. For example, ammonia, all those species which contain lone pair of electron will have the tendency to be protonated by the proton, hence they will attract the proton towards itself, and hence they will be acting as proton acceptor. So next is, in order to perform feasible titrations of weak bases, the solvent system should be selected specifically in such a fashion so as to eliminate as far as possible the competing reaction of the water for the proton, besides enhancing the strength of the basic species. So in order to perform feasible titrations of weak bases, the solvent should be selected in such a manner so that we can eliminate the competing reactions of water as far as possible. We should try to select the solvent so that we can completely finish off or eliminate the competing reaction of water for the proton, besides enhancing the strength of the basic species. Now, before we discuss more about non-aqueous titrations, we will study the different types of non-aqueous solvents which we use in the non-aqueous titrations. So, the first one is aprotic solvents. Aprotic solvents are generally non-polar. They are neutral. They do not have any charge. The atoms present in the aprotic solvents have similar electronegativities, and because of the similar electronegativities, there will be no charge on the solvent, and hence our solvent will be acting as a neutral solvent. Since they do not have any positive or negative charge, they will be chemically inert, and also they will be having low dielectric constant because there are no positive negative ends in the solvents. There is no formation of positive and negative charge in the solvent, hence they will not dissociate themselves into solvent very easily, hence the dielectric constant of aprotic solvents is very low. Examples of these types of solvents include chloroform, benzene. So the examples which come under aprotic solvents is that of chloroform and benzene. In chloroform, since we know that we have carbon, we have hydrogen atoms, electronegativity difference is not that high, so it acts as non-polar solvent. In benzene, same way, it is organic compound, it will not be acting as a protic solvent, it will be completely neutral, it has no charge, and hence it will act as aprotic solvent. Chloroform, although because of the presence of two chlorine groups, it will have slight polar character as compared to benzene. Benzene is completely neutral, it is completely non-polar molecule. There is no formation of positive or negative charges in benzene, whereas in case of chloroform, because of the presence of three chlorine atoms in it, there is slight polarity in the chloroform, but the non-polar character of chloroform dominates as compared to the polar form, whereas in benzene, because there are only carbon and hydrogens present in it, it is almost completely neutral. It is almost completely neutral. And also aprotic solvents, they are not, since they are non-polar, they have no tendency to release protons when dissolved in aqueous medium. Since they are non-polar, they have no tendency to release proton ions when dissociated when dissolved in the aqueous medium. Hence they are known as aprotic solvents, because they are not capable of releasing proton ions in the aqueous medium. In the solution phase, they will not be able to release any proton ions. And the next solvent is protophilic solvents. What are protophilic solvents? Protophilic solvents are those which are proton loving. They are basic in character. They might be having electron density on them, and hence they have tendency to attract the proton towards themselves. Hence they are known as protophilic solvents. These solvents have a basic reactor and tend to react with the acids when come in contact with, leading to the formation of solvated protons. So or in what happens in case of protic solvents, because of the basic reactor, due to the lone pair of electrons present in them, due to the electron charge present on them, due to the electron charge present on them, they have tendency to attract the proton towards themselves. And when the protons attract with the electrons, they solvate that molecule, solvate that solvent, and hence lead to the formation of solvated protons. Now, examples of protophilic solvents are ammonia. Ammonia acts as protophilic solvent because of the presence of lone pair. The lone pair is going to attract the proton towards itself. Hence, it is known as protophilic solvent. Second is pyridine. In pyridine also we have a lone pair of electron. The lone pair of electron is easily available for protonation, and hence it acts as protophilic solvent. The third solvent is protogenic solvents. Protogenic solvents are they show acidic character. The protogenic solvents are those which themselves are acidic. They have tendency to release H positive ions. They have tendency to release proton ions when dissolved in the solvent. So these solvents have a more acidic character and tend to have a leveling effect on the bases they come in contact with. Now what do you mean by leveling effect of bases? In protogenic solvents, basically we include those solvents which have much more acidic character. Much more acidic character means that it has tendency to release H positive ions very quickly. So if it is going to release H positive ions very quickly, the H positive ions will be accepted by weak base also and a strong base also, because protogenic solvents being very acidic in character, they have tendency to give H positive ions to weak base as well as to strong base. So hence they act as leveling, they show leveling effect, and hence they act as leveling solvents. So in protogenic solvents, these solvents, because of their high acidic character, they tend to have a leveling effect. They show leveling effect. It means that all the bases, be it weak base or a strong base, is going to accept the proton. It is going to accept the proton which is released by the protogenic solvents, and hence they are, we can say that they are on the same level, and hence protogenic solvents are known as leveling solvents, because they keep all the bases on the same level, be it a weak base, be it a strong base. The protogenic solvent is going to give proton to weak base as well as strong base. So hence it is known as leveling solvent also. Protogenic solvents are hence known as leveling solvents also. Examples of protogenic solvents which are used in non-aqueous titrations are sulfuric acid and acetic acid. Amphiprotic solvents. What are amphiprotic solvents? These are those solvents which have the properties of both protophilic as well as protogenic solvents. So amphiprotic solvents are those which have the characteristics of both both the protophilic as well as protogenic solvents, and the examples include acetic acid. Acetic acid acts as amphiprotic solvents because acetic acid contains a alkyl chain also. Because of the presence this alkyl chain, because it consists of only carbon and hydrogen, it is protophilic in nature. It is non-polar. It does not act as polar compound. Because of only carbon and hydrogen, it is that portion is completely organic, so it is acting as completely non-polar part. Whereas the hydroxyl group which we have is polar in nature because of the presence of OH group, which is polar. We know that OH group has polarity because of the electronegativity difference of oxygen and hydrogen. Because of the electronegativity difference between oxygen and hydrogen, these solvents will be acting as amphiprotic solvents because they have non-polar portion also. With that, they have a polar portion also. So next topic is alkymetry and acidimetry titrations. These titrations are very very important for the quantitative estimation of basic and acidic drugs. What happens in case of acidimetric titrations? In acidimetric titrations, the quantitative estimation of the basic drug is done with the use of acidic standard solution. So what happens in acidimetry titrations? In acidimetry titrations, we are going to do the estimation of opposite to acid, that is a basic drug, but the titrant is going to be acidic in nature. For example, normally we use perchloric acid as the titrant in acidimetry titrations. The perchloric acid will be acting as the standard solution, and it will help us to do the quantitative estimation of the basic drug. Now, protogenic solvents are used in acidimetry. Since we are going to find out the quantitative, we are going to find out the quantitative estimation of the basic drug. So for this, we will be using the titrant which is acidic in nature. In acidimetry, we will be using those solvents which are proton releaser, which have tendency to release protons. All those solvents are known as protogenic solvents, like glacial acetic acid, which has tendency to release H positive ions, can be used in acidimetry titrations. And various samples which can be determined by the acidimetry are ephedrine, morphine, caffeine. All those drugs which are basic in nature can be quantitatively estimated by using acidimetry titrations. Other drugs such as adrenaline, all those which are basic in nature, maybe because of the presence of the lone pair, can be quantitatively estimated by using acidimetric titrations. The indicator which is normally used in acidimetry titrations is crystal violet. Crystal violet indicator is used while performing the acidimetry titrations. Now the titration of a weak base by non-aqueous titration. In the titration of a weak base by the non-aqueous titration, we will have to consider a few points. First is the titrant use. What is the titrant which we are using? For the estimation of a weak base, the titrant used in the acidimetry will be a protogenic solvent which has acidic character. Titrant use can be perchloric acid also. So the next step is the preparation of perchloric acid, that is preparation of 0.1 normal perchloric acid, and then we standardize it so that by standardization we mean that we check that the concentration should be exactly 0.1 normal, it should not vary. And by standardization we can prepare a standard solution. Standard solution means that by perchloric acid, by preparing the standard solution of perchloric acid, we can find out the concentration of another unknown sample, unknown base drug, which we will be using. So the standardization is very important. The third point is the solvent use. We will also be using a solvent, and the solvents which are generally used are free from moisture, free from any type of water content, because so far we have studied that water competes with the reaction. It produces competing reactions which are undesirable, which may result in error, and hence the solvent which we must be using should be free from any moisture or water content. Fourth is the practical examples of weak bases along with the indicators. So we will also be considering the practical examples of the weak bases along with the indicators. And as we have discussed that we by acidimetric titrations, we can find out the we can do the quantitative estimation of weakly basic substances, like example ephedrine. What is alkymetry titration? Exactly the opposite of acidimetry titration. In alkymetry titration, we are going to perform the quantitative estimation. We are going to find the quantitative estimation of the weak basic drugs. So in the alkymetry, we are going to perform the quantitative estimation of weak acidic drug. The titrant which we will be using in the alkymetry titrations will be basic or alkaline in nature. So here the quantitative estimation of weak acidic drug will be done in alkymetry, but the titrant used will be basic or alkaline in nature. Example of the titrant which we can use in alkymetry titration is sodium methoxide. Here we can see that methoxide group is joined by the sodium ion. It is surrounded by the sodium ion. Now the protophilic solvents which are used in alkymetry titrations are dimethyl formamide, which is known as DMF. It is basically the derivative of amine. Amide is a derivative of amine, and it has been substituted. The nitrogen in the nitrogen of the amide group has been substituted by two methyl groups. The samples which can be determined by the alkymetry titrations include weak acidic drugs, which are given here, and the indicator which is used in the alkymetry titrations is thymol blue. Thymol blue. Now we will be studying the exact procedure of the non-aqueous titrations. NAT refers to non-aqueous titrations. Now we will be studying that in more detail. In the non-aqueous titrations of the weakly basic drug, we will be doing the titration against perchloric acid. Now, most of the drugs which we which we consume are either weakly acidic or basic in nature. So the drugs are weakly acidic in or basic in nature. For the estimation of these drugs, these are dissolved in suitable solvent. For the estimation of the drugs, we have to dissolve them in a suitable solvent, and then titrate it against suitable titrant. In case of weakly basic drugs, we dissolve it in glacial acetic acid and titrate with perchloric acid. So normally for the quantitative estimation of the weakly basic drug, we dissolve it in glacial acetic acid, acts as a solvent, and we titrate it with perchloric acid. Now, what does glacial acetic acid does? Glacial acetic acid is going to dissociate itself into proton ions and acetate ion. But when strong acid, now perchloric acid is much strong acid as compared to acetic acid. But when strong acid perchloric acid, that is perchloric acid HClO4, is added to glacial acetic acid, there is the formation of onium ions. We will be seeing that glacial acetic acid, now here being a weak acid as compared to perchloric acid, will act as a base, and perchloric acid is going to protonate the acetic acid. On protonation, acetic acid gets converted into a species here, which we can see that is onium ion CH3COOH2 positive. Now this acetic acid, due to the formation of onium ion, behaves as a strong acid. Now what happens here is, now what has happened, acetic acid, which was a weak acid before, because after the protonation, after the acceptance of the proton by the perchloric acid, will act as a strong acid due to the formation of onium ion. So here we can see, earlier acetic acid was a weak acid, but after dissolving it in perchloric acid is very strong acid. It is one of the strongest acid. It releases H positive ion very fastly, and this H positive ion is going to combine with glacial acetic acid, and hence there is the formation of onium ion. This onium ion is very strong acid, and it releases H positive ion very rapidly into the medium. Now, when we use weakly basic substance such as pyridine, when we add weakly basic substance to pyridine, to the when weakly basic substance such as pyridine, we can use any other, we can estimate any other basic drug we want to estimate. So when we add any basic substance, for example, I have taken here pyridine. When I dissolve pyridine in acetic acid, equivalent amount of acetate ions are produced. So according to this well balanced chemical equation, 1 mole of pyridine is going to combine with, it is going to react with 1 mole of acetic acid, and there will be formation of equivalent amount of acetate ion with the protonated pyridine ion. So what has happened here, the H positive from the acetic acid has been used for the protonation of the lone pair present on the nitrogen, and acetate ion has been produced. Hence, this will lead to increased tendency to accept protons. Now, what has happened here? Hence, this is going to lead to the increased tendency to accept protons because of the formation of acetate ion. This acetate ion is going to accept, because we know that acetic acid is a weak acid. So acetic acid, after the removal of H positive ion, is going to form a conjugate base. Although acetic acid is a very weak acid, but after the removal of H positive ion, according to the acid base conjugate concept, acetic acid, after the removal of H positive ion, will form carboxylate ion, acetate ion, which will act as a very strong base now. So this acetate ion which has been produced will increase the tendency to accept the proton. Hence, this mixture will act as a strong base. Because of the formation of the acetate ion, this acetate ion acts as a very strong base. And because of the formation of the acetate ion, this mixture is going to act as a strong base. Now, what are the various reactions which will take place here? First of all, the perchloric acid, which we are using as a titrant here. First of all, it is going to protonate the acetic acid, acetic acid is the solvent which we are using here. So acetic acid is acting as a solvent here also, because acetic acid is a weak acid as compared to perchloric acid. So the perchloric acid is going to release H positive ions. The H positive ions will protonate the acetic acid, and there is formation of onium ion. So this is the equation number 1. In the equation number 2, any drug which we want to estimate, any weak basic drug, for example, I have taken here pyridine. Pyridine, because of the presence of lone pair, acts as a base, as we all know. It is going to react with acetic acid, and the acetic acid, after it is going to release H positive ion, and that H positive ion is going to protonate the lone pair present on the pyridine. It is going to protonate the lone pair of electrons present on the pyridine. So hence we get an acetate ion here. So these are the two equations. First of all, first equation is the reaction of perchloric acid with acetic acid, and the second one is the reaction of pyridine with acetic acid. Now we can see that we have got onium ion from equation 1, we have got an acetate ion from equation 2, these two ions, the onium ion and the acetate ion which we have got, will combine, will react to form acetic acid. Sir? So the onium ion and the acetate ion which we have produced will react. So the onium ion and the acetate ion which we have produced, they will react to form molecules of acetic acid. Hence, we can say that indirectly complete neutralization of the reaction has taken place, and after the complete neutralization end point is obtained, the net reaction is written as perchloric acid reacts with pyridine to form ClO4 negative, that is the ion which we have got from perchloric acid and the protonated pyridine. So this is the net reaction. Here glacial acetic acid is acting as a solvent, and it has helped to complete the reaction, to complete the neutralization reaction. Now, this was the main theory behind the titration, that was acidimetry titration. Now, we will find out the different solutions. We will study the different procedures which will be used for the preparation of the different reagents which we are using in this titration. First of all, first preparation is the preparation of 0.1 normal perchloric acid. So for the preparation of 0.1 normal perchloric acid, we will be using glacial acetic acid as we will be using acetic acid as the solvent. So first of all, we will be taking 500 ml of anhydrous glacial acetic acid. It must be kept in mind that always anhydrous glacial acetic acid will be used in the non-aqueous titrations, as we have studied that because of any water content or moisture in any solvent, it may compete in the reaction and hence result in an undesirable reaction which may produce an error. So first of all, we are to take about 500 ml of anhydrous glacial acetic acid and then add 25 ml of acetic anhydride in it. The flask in which we are taking anhydrous glacial acetic acid and acetic anhydride, it should be completely cleaned and dried. There should be no water droplets in the volumetric flask. What is the purpose of adding acetic anhydride in it? The purpose of adding acetic anhydride is the water which is present in the solution or on the flask will react with acetic anhydride to produce acetic acid. Acetic anhydride will help us to make sure that there is no water content left in the volumetric flask. So this is the purpose of using acetic anhydride. Any water droplets, any moisture which is present in the volumetric flask will react with acetic anhydride to ultimately give us acetic acid. Second, we have prepared our solvent and we have completely made it water free. Now we will be adding perchloric acid in it. Now add about 8.5 ml of perchloric acid, about 70% with continuous stirring. So continuous stirring is done to prepare an homogeneous mixture for titration. Always we should be sure that the solution which we are using should be homogeneous. If our solution is heterogeneous, it will settle at the bottom, and hence our solution will not be having uniform concentration throughout the flask. The upper layer might be having lower concentration and the lower layer might be having higher concentration. So the mixing stirring should be done it should be done at a proper interval so that the solution prepared should be completely uniform concentration. Now, after the addition of perchloric acid in glacial acetic acid, we will cool the solution and we will make up the volume till 1000 ml with anhydrous glacial acetic acid. We will be adding 500 ml of anhydrous glacial acetic acid with the addition of 25 ml of acetic acid and 8.5 ml perchloric acid. After cooling the mixture, we will make the volume up to 1000 ml. Again, we will mix the solution thoroughly. Now we will not be using this solution immediately for the titration. We will keep the solution for at least 24 hours so that any excess acetic anhydride which is present in our solution can be combined, so that all the acetic anhydride can combine with any water droplets, any moisture which is present in our glacial acetic acid. Then carry out the determination for water. If the water, okay, now to make sure that our solvent, the titrant which we are using, that is perchloric acetic acid, is free from any kind of moisture, we will carry out the determination of water also. If the water content is more than 0.5%, we will add more acetic acid so that the water content is completely finished. So we will be adding more acetic anhydride if there is still water content more than 0.5%. Now, after keeping the solution for 24 hours, we will allow the solution to stand for 1 day, and again we will titrate the water content. We will again find out the water content. If the water content again comes out to be more, we will have to add more acetic anhydride and keep it for one another day, so that the acetic anhydride which we have added will combine with the water molecules present in the medium. The solution so obtained should contain the water between 0 to 0.0% and 0.05%. The water content should not be more than that, otherwise it will result into undesirable error. And the next one is the standardization of 0.1 normal perchloric acid. Now we will find out the solution which we have prepared for of 0.1 normal perchloric acid. Is it standard or not? For that we will be doing this following steps. We will weigh accurately 0.7 gram of potassium hydrogen phthalate. We will powder it completely and we will dry it for 2 hours. Drying purpose is again done so that our sample is free from any type of water content. We will dissolve it in 50 ml of glacial acetic acid. Then we will be adding few drops of indicator. The indicator which is used here is crystal violet, and we will titrate it with perchloric acid. We will titrate it with the perchloric acid till violet color changes to emerald green. So we will be performing the titration till the color changes from the violet type color to the greenish type color. So nextly we will be performing the blank titration by using 50 ml. Here in the blank titration we will not be using the potassium hydrogen phthalate. We will be performing the blank titration by using 50 ml of the glacial acetic acid, and we will subtract the volume of perchloric acid which is consumed from the reading which we have got above. For the standardization, we will perform this procedure. First of all, we will be carrying out the main reaction. Secondly, we will perform the blank titration. The volume which has been used in the first case, the volume of the perchloric acid which has been used in first case, we are going to subtract that. We will be subtracting from that value the second volume which we will be getting after in the second step, that is the blank titration. From that we will come to know about the strength of the perchloric acid which we are going to use. Now what are the various equations, what are the various reactions which will be involved, what are the various numerical values which are involved in the standardization of 0.1 normal perchloric acid. Each ml of 0.1 normal perchloric acid is equal to 0.02042 gram of potassium hydrogen phthalate. So I want to say that each ml of zero point 0.1 normal perchloric acid is equal to 0.02042 grams of potassium hydrogen phthalate. So what will be the strength? How we find out the strength? We find out the strength of the perchloric acid by dividing the weight of the potassium hydrogen phthalate with the volume of perchloric acid which we have used, and also by the also by the value of the potassium hydrogen phthalate, which is equal to 1 ml of the zero point 0.1 normal perchloric acid, that is 0.02042. So from this we will be getting that 204.2 gram of potassium hydrogen phthalate is equal to the 1000 ml in perchloric acid. So 0.00242 gram of potassium hydrogen phthalate will be equal to, this is done by simple unitary method, it comes out to be 1 ml in perchloric acid. Now, this is the reaction which has been taking place in the standardization of zero point 0.1 normal perchloric acid. Potassium hydrogen phthalate is going to react with perchloric acid to form phthalic acid, and with the release of ion, and with the release of potassium acid. So what are we getting here? By this equation, by this standardization, we can find out the strength of the perchloric acid very accurately. So this was all about the ultimate. This was all about the acidimetric titrations. In acidimetry titrations, what we have studied, acidimetry titration is used for the estimation of quantitative estimation of weak base. We will be using here the titrant will be acidic in nature. So what we have done here, we have used solvent. We have used acetic acid as a solvent, as a medium of the solvent, and the base here which we have used is that of pyridine. We are using pyridine, and the titrant here is being the titrant used here is the titrant which we are using here is perchloric acid. So what we have observed here is, when we will dissolve acetic acid in perchloric acid, acetic acid being a weak acid is going to be protonated by the perchloric acid which we have added. It is going to protonate the acetic acid. It was going to result into the formation of onium ion, and the pyridine, that is the sample which we want to estimate the we will be, it is also going to react with the acetic acid to form acetate ion, and protonated pyridine is going to be formed. So these are the two reactions which will take place. In both the reactions, we see that in the first reaction onium ion has been formed, which act as a acid, a very strong acid, and acetate ion has been formed which acts as a very base. These two ions are going to combine and they are going to form acetic acid. This complete neutralization takes place and the endpoint is very sharp. Nextly, we have studied the preparation of perchloric acid. We have studied that perchloric acid solution was prepared in glacial acetic acid. To make sure that no water content is present in it, we have added acetic anhydride into it, and we have kept it for a single day so that all the acetic anhydride can be consumed by reacting with the water molecules. So, and while preparing zero point 0.1 normal perchloric acid, we should keep in mind, we should be very sure that the water content should not be more than 0.5%. We should be very sure about this, because the water content which will be present in the flask will interfere with the reaction. Next was the standardization of zero point 0.1 normal. We have done the standardization of zero point 0.1 normal perchloric acid by titrating it with potassium hydrogen phthalate. Potassium hydrogen phthalate reacts with the perchloric acid and forms phthalic acid. By using this equation, we can find out the strength of the perchloric acid which is being used, and we can accurately find out we can accurately standardize our solution. So each ml of zero point 0.1 normal perchloric acid we have studied it is equal to 0.02042 grams of potassium hydrogen phthalate. So from this formula we can calculate the strength of the perchloric acid very easily. So the next one is the non-aqueous titrations of the weakly acidic drug. Now we will be performing the non-aqueous titrations of the weakly acidic drug. Earlier we have performed the non-aqueous titrations of weakly basic drug. Now we will be performing the non-aqueous titrations of the weakly acidic drug. Titrations of weak acidic drugs such as barbiturates, and benzoic acids can be done by using bases such as alkali methoxides. Here, the titrant acting will be alkali methoxides. The non-aqueous titrations of weakly as basic drug, we have used perchloric acid. Here we will be using alkali methoxide as titrant. We can use sodium methoxide, we can use potassium methoxide, we can use lithium methoxide or tetrabutyl ammonium hydroxide. All these titrants can be used in the non-aqueous titrations of the weakly acidic drug. Non-aqueous solvents used in the titrations are the solvents which we will be using in non-aqueous titrations is mostly dimethyl formamide, that is DMF, but ethylene diamine is also used commonly in the non-aqueous titrations. Reactions taking place during the titration of benzoic acid in DMF are as follows. Now we will be studying all the reactions which takes place during the non-aqueous titrations of the weakly acidic drug. First of all, for example, we are performing the quantitative estimation of benzoic acid. When we will be dissolving benzoic acid in our solvent, which is DMF here, that is dimethyl formamide, what happens? The benzoic acid is going to release H positive ion and it is going to protonate the electrons, the lone pair of electrons present on the amide group. So the electrons will be protonated by the proton which is released by the benzoic acid. Secondly, the titrant which we are using, that is sodium methoxide, will dissociate into sodium ions and methoxide ions. From these two equations we have the protonated DMF and the the methoxy ion which has been produced in the step number 2. They are going to react and they are going to result into the formation of DMF and alcohol. Now now what is the net reaction? In the net reaction we can see that acetic acid reacts with sodium methoxide and forms salt of benzoic acid with the release of alcohol, with the release of methanol. So this is the net reaction, this is the net neutralization reaction which is taking place here. What happens in the reaction? Initially the benzoic acid is going to react with DMF, the proton is going to protonate the amide group and carboxylate ion, that is the benzoate ion, is formed. Secondly, in the second step, ionization of sodium methoxide ion take place. Sodium methoxide ion dissociates itself into sodium ions and methoxide ions. From step number 1 and step number 2 we can see that the protonated DMF and the methoxide ion which has been produced in step number 2, they are going to combine and result into the formation of DMF and alcohol. And the last step is the overall reaction. In the overall reaction we can see that these two species have combined, so we will not be writing these two species, we will be writing these two. The benzoic acid reacts with sodium methoxide, and it results into the formation of the salt and alcohol. So this is the net overall reaction which we have written. We have skipped these two portions and instead of these two portions we have written DMF and alcohol. In overall reaction, we can see that in overall reaction there is no use of the solvent, but we can observe that the solvent has played his role in the internal reaction. Non-aqueous titration of weakly acidic drug. Now we will be studying the different solutions which are prepared during the non-aqueous titrations of the weakly acidic drug. We will be preparing the solutions, then we will be standardizing the solutions. First of all, we will be preparing the solution of zero point 0.1 normal sodium methoxide solution. So how is the preparation of zero point 0.1 normal sodium methoxide solution done? We will add 2.5 grams of freshly cut sodium metal in 150 ml of methanol. So we will be adding 2.5 gram freshly cut. The sodium metal which will be used should be freshly cut, because if it is not freshly cut, there are possibilities that the sodium metal might have reacted with the oxygen and might have get oxidized, and there might be a layer of there might be oxide layer on the sodium metal surface. So the sodium metal which we are going to use should be freshly cut, so that it should react easily, because the if it is not freshly cut, it might contain the oxide layer of the sodium oxide layer. So add 2.5 grams of freshly cut sodium metal in 150 ml of methanol. When it dissolves completely, dilute it with dry toluene up to 1000 ml. So first of all, how we will be preparing the zero point 0.1 normal sodium methoxide solution? We will cut freshly sodium metal. The quantity will be 2.5 gram. We will put it into 150 ml of methanol. We will dissolve it completely. When it has completely dissolved in the methanol, we will dilute it with dry toluene up to 1000 ml. The next step is the standardization. How we will be performing the standardization? The standardization of the sodium methoxide ion produced will be done in the following way. First step is we will be adding 0.4 gram benzoic acid in 80 ml of DMF, and into that solution we will be adding 3 drops of thymol blue indicator. So first step is going to be addition of benzoic acid into the solvent, which is DMF here, with the addition of indicator, that is thymol blue. 3 drops of indicator will be added. Then we will be titrating the mixture. We will titrate the mixture against sodium methoxide solution to obtain blue end point. Then we will be, this is the first main titration. We will be performing the blank titration by skipping benzoic acid. We will not be adding benzoic acid in the blank titration. Perform the blank titration by omitting the benzoic acid, and then by the volume of the sodium methoxide which has been used in the first step, and the amount and the volume of the sodium methoxide which is being used in the second step, we are going to subtract, and by the volume difference we can calculate the strength, the accurate strength of the sodium methoxide solution. Factor each 1 ml. So this is the important point which we must keep in our mind, that 1 ml of 0.1 normal sodium methoxide. 1 ml of 0.1 normal sodium methoxide is equal to 0.01221 gram of benzoic acid. So according to this equation, 1 ml of 0.1 normal sodium methoxide is equal to 0.01221 gram of benzoic acid. So by using this equation, we can find exactly the strength of the sodium methoxide used. By finding out how much sodium methoxide has been used for the neutralization of 0.4 gram benzoic acid, we can calculate the strength of the sodium methoxide very easily. The procedure for weakly acidic drug quantitative estimation is: first, dissolve weakly acidic drug in the solvent. First of all, we will dissolve the weakly acidic drug whose quantitative analysis is to be done. We will dissolve it in the suitable solvent, which can be toluene, which can be methanol, which can be DMF. After dissolving the weakly acidic drug completely in the solvent, we will be adding 2 to 3 drops of suitable indicator. The choice of indicator will be based on the drug, the acidic drug which we are taking. So we will be adding 2 to 3 drops of suitable indicator. Now nextly we will fill the burette. We will fill the burette with the sodium methoxide ion or potassium methoxide ion. The main role played here will be by the methoxide ion. Now we will titrate the contents of the flask against the titrant till we get endpoint. Now we are going to perform the titration till the endpoint is observed. After that, we will also be performing the blank titration. In the blank titration, we will not be using a weakly acidic drug. We will not be using the drug. In the blank titration, we will skip the addition of weakly acidic drug and we will get the exact amount of the titrant which is required for the quantitative estimation of which is used to completely react with the weakly acidic drug. So the examples of drugs that can be titrated, these are the few examples of the drugs which can be estimated by non-aqueous titrations, by alkymetric titrations, and these are the various names of the indicators used. So here we can see that we have first of all dissolved the drug in the solvent, we have added the indicator, and titrate, we will titrate it with the sodium methoxide solution which we have prepared, which we have standardized by reacting it with benzoic acid. Now, how is the preparation of zero point 0.1 normal lithium methoxide done? We have studied the standardization, the preparation of sodium methoxide solution. Now we will be studying the preparation of zero point 0.1 normal lithium methoxide solution. So how is the preparation done? The preparation of zero point 0.1 normal lithium methoxide solution is done by the following steps. First of all, add 0.7 gram of freshly cut lithium in a mixture of 40 ml of methanol, and then we have to add 50 ml of dry toluene into it. Repeat the addition of methanol and toluene in the similar ratio till clear solution is obtained. So again, we will repeat the addition again with the methanol and toluene now in the same ratio till we will be continuing the addition till the clear solution is obtained. Then we are going to make up the solution up to 1000 ml by the addition of toluene. Standardization of lithium methoxide is done in the similar method as we have done for sodium methoxide. Examples of the drugs which can be titrated against lithium methoxide are as follows. Allopurinol can be estimated by using 0.1 normal lithium methoxide solution as a titrant. The indicator used is thymol blue. Mercaptopurine can also be estimated. The quantitative estimation can be done by using 0.1 normal lithium methoxide solution, and the indicator will be the same, thymol blue. What are the advantages of non-aqueous titrations? This was all about the alkymetry titrations. We have studied different solvents which are used in both acidimetry and alkymetry titrations. We have studied the titrants which can be used. Now we will study why is there a need for what are the advantages of non-aqueous titrations over those of aqueous titrations. So the first advantage is that organic acids and bases that are insoluble in water are soluble in non-aqueous titrations. We know that organic acids and bases, they do not dissolve, they are immiscible, they are insoluble in water, and hence we cannot perform the titrations of organic compounds in water, because since the organic acids or bases, they are not going to completely dissolve them in water. We cannot get a homogeneous solution. If you cannot get a homogeneous solution, you cannot get a solution of that organic acid or base, then the titration is not possible, because we know that for the performation of titration, for performing titration, there is always the need for homogeneous solutions. So second point is organic acid, which is of comparable strength to water, can be titrated easily in non-aqueous solvent. Bases also follow the same rules. Now the next problem was that organic acids, as we know that organic acids are not very acidic, as acidic as inorganic acids like sulfuric acid, hydrochloric acid, or nitric acid. Their acidic strength is comparable to that of water. So if their acidic strength is comparable to that of water, they cannot be estimated by aqueous titrations. Hence, there is need for performing non-aqueous titrations. Third advantage of non-aqueous titration is that a non-aqueous solvent may help two or more acids in the mixture. The individual acid can give separate endpoint in different solvent. Another very important advantage of using non-aqueous solvents is that in the non-aqueous titrations we can perform the titrations of two or more acids which are going to separate, which are going to give different endpoints in the different solvents. So by non-aqueous titrations, by using non-aqueous solvents, we can find out the endpoint of more than one acids. The fourth advantage of non-aqueous titrations is that by the proper choice of solvents of indicator, by the proper choice of the solvents and indicator, the biological ingredients of the substance which are acidic or basic in nature can also be easily titrated. So the another important advantage is that estimation of biological ingredients of a substance who are acidic or basic in nature can also be done by proper choice of solvents or indicators. The fifth point is non-aqueous titrations are very simple and they are very accurate. The examples of non-aqueous titrations are ephedrine preparation, codeine, phosphate in APC, and many more. Now, next topic is, this is all about the alkymetry and acidimetry titrations. The next part of the unit, the last part of the unit, is estimation of sodium benzoate by non-aqueous titrations. So this is a very important topic. How is the estimation of sodium benzoate done? We will study the procedure for that. Before that, we will discuss the properties of sodium benzoate. This is the structure of sodium benzoate. The structure of in the structure of sodium benzoate, we can see that benzene is being attached to the benzoate group is attached to the sodium ion. So here we have a carboxylate group. The carboxylate group is attached to sodium ion. The molecular formula, the molecular weight of sodium benzoate is 144.1 gram, and the chemical formula is C7. There are 7 carbon atoms and 5 hydrogen atoms, and with 2 oxygen atoms. Sodium benzoate is white in color. It is crystalline or in the granule form. It is slightly hygroscopic. Hygroscopic means it is moisture sensitive. It has tendency to absorb moisture. It is slightly hygroscopic and it is freely soluble in water. It is freely soluble in water because of the presence of this carboxylate group. It is freely soluble in water and it is also springly soluble in alcohol. Why is it springly soluble in alcohol? It is springly soluble in alcohol because of the presence of this benzene ring, because of the dual character, because of the presence of polar as well as non-polar portion. It is freely soluble in water as well as it is springly soluble in alcohol. The powder of sodium benzoate, when it is dried, contains not less than 99% of sodium benzoate. So when the powder of the sodium benzoate is dried, it does not contain less than 99% of the sodium benzoate. Sodium benzoate has antibacterial and antifungal properties, and it is therefore used. It is also used as a preservative in the pharmaceutical formulations, including oral preparations. Its concentration as a preservative in the pharmaceutical formulations is up to 0.5%. Sodium benzoate is also common ingredient of cough preparations. So sodium benzoate is also used as a common ingredient in the cough preparations. So these are the properties of sodium benzoate: the molecular weight, the molecular formula. The color is white. It is in the crystalline or the granule form. It is slightly hygroscopic. It is moisture sensitive. It is freely soluble in the water because of the benzene, because of the presence of carboxylate group, and it is springly soluble in water because of the benzene ring. And also it has antibacterial and antifungal properties. It is used as a preservative in the pharmaceutical formulations, including oral preparations, and its concentration as a preservative is up to 0.5%. Next is the principle which is involved in the estimation of sodium benzoate by non-aqueous titrations. The principle is that sodium benzoate is a base. We know that sodium benzoate acts as a base. When it is dissolved in glacial acetic acid, it is dissolved in glacial acetic acid. Sodium acid, so the solution of the sodium benzoate is prepared by dissolving it in glacial acetic acid. During the titration, when the titration of the sodium benzoate is done by using strong acid as a titrant, we have studied this theory before. Acetic acid behaves like a base. Acetic acid is going to behave like a base, and the accurate endpoint is determined by using suitable indicator. Endpoint is detected by using 1-naphthol benzene as an indicator. So the principle is exactly the same as the acidometric titration which we have studied. Now we will be discussing the preparation and the standardization of perchloric acid which will be used here. First of all, we have to dissolve 8.5 ml of perchloric acid, that is approximately 72%, in 900 ml of glacial acetic acid with continuous stirring. Continuous stirring must be done while preparing any solution. And then we will be adding 30 ml acetic anhydride into it. Again, acetic anhydride is done so that all the water droplets, all the water content which is present in glacial acetic acid or perchloric acid can be consumed by reacting with acetic anhydride. On the reaction of acetic anhydride with water, we will be getting again acetic acid which will act as a solvent. And then after the addition of acetic anhydride, we will make the volume up to 1000 ml with the further more addition of glacial acetic acid. We keep this flask for 24 hours so that complete reaction can take place. All the acetic anhydride which we have added can be consumed by reacting with water. After 24 hours, when acetic anhydride has completely absorbed all the water from perchloric acid and glacial acetic acid and renders the solution virtually anhydrous. After 24 hours, it is approximately estimated that our solution is completely free from water and it is in complete anhydrous form. Perchloric acid must be well diluted with the glacial acetic acid before adding acetic anhydride, because reaction between perchloric acid and acetic anhydride is also explosive. One important point which must be kept in mind before addition of acetic anhydride into perchloric acid is that before the addition of acetic anhydride, the sample of perchloric acid must be diluted with glacial acetic acid, because the direct reaction of perchloric acid with the acetic anhydride can be very explosive. To avoid that explosive reaction, first of all, the perchloric acid solution should be diluted with the glacial acetic acid, and then the addition of acetic anhydride must be done. Now, after the preparation of the perchloric acid solution, its standardization is very important. So, how is the standardization of perchloric acid done? Use 500 mg of potassium acid perthalate. It is the same, exactly to the same which we have discussed in acidimetric titrations. We will be taking approximately 0.5 gram of potassium acid per phthalate. To that we will add 25 ml of glacial acetic acid. Glacial acetic acid acts as a solvent. Here we will be adding few drops of 5% weight by volume crystal violet glacial acetic as indicator. So crystal violet is used here as an indicator for the standardization of perchloric acid, and the indicator should be prepared in glacial acetic acid. This solution is titrated with 0.5 perchloric acid till the endpoint is observed. The endpoint is again the same from violet to green color. Now we will study the procedure for the estimation of sodium benzoate. Firstly, we have to accurately weigh 0.25 gram of sodium benzoate. Sodium benzoate is we are performing the estimation of sodium benzoate. So we are to accurately weigh 0.25 gram of sodium benzoate, and we will transfer it to the conical flask where the titration will be done. Then we will add 20 ml of anhydrous glacial acetic acid into it, and we will dissolve it with the help of sonicator, and we can also heat it. We can just warm the solution if required for the dissolution of sodium benzoate into the glacial acetic acid. After that, after the preparation of the sodium benzoate solution, complete formation of the solution, we will be adding few drops of indicator. We will add 2 drops of 1-naphthol benzene indicator solution. After the addition of indicator, we will start with the titration. For the performation of titration, we will fill the burette with the standardized solution of perchloric acid. The standard solution of perchloric acid which we have prepared, we will fill that in the burette, and we will fill that in the burette. Before filling perchloric acid in the burette, we should be sure that burette is cleaned with water, it is rinsed with water, and also rinsed with perchloric acid. After the filling of burette with the perchloric acid solution, we will start with the titration. Start the titration with the perchloric acid till the end point is reached. So we will start the titration and till the end point is reached, and we will record the reading of the perchloric acid which is being used for the neutralization to react with the sodium benzoate solution which we have taken in the flask. Now the estimation of sodium benzoate by non-aqueous titrations. Now there are some calculations which we will be using for the estimation of the sodium benzoate by non-aqueous titrations. We will not be discussing the complete calculations. We will just be discussing the result. The percentage of sodium benzoate which is present in the solution can be calculated by XML. XML is the ml of perchloric acid which has been used for the reaction of sodium benzoate. The XML will come after performing the titration which we have discussed now, and after performing the blank titration. For the performation of blank titration, we will not be adding sodium benzoate. So first of all, we will be performing the reaction with the addition of sodium benzoate. That is our main reaction. That is our main titration. After that reaction, after the titration, we will be performing blank titration. In blank titration, we will not be using sodium benzoate. In the first step, we will calculate the volume which has been required for the volume of the perchloric acid which has been required for the estimation of sodium benzoate, and then we will be calculating the volume of perchloric acid used for the second titration. We will subtract the volume of perchloric acid for both the titrations, and that will come out to be the XML here. We will substitute that value here. We will multiply it by the normality, and we will multiply it by 0.01441 factor and multiplied by 100. N of perchloric acid is known to us and the weight of the sample is also known to us. The equivalent factor here we have used is 0.01441 gram. Why this vector has come here? It has come from here. This is the equivalent factor of the sodium benzoate for 0.1 normal perchloric acid solution. It is approximately 0.01441 gram. Here X represents the volume of titrant which has been consumed for the endpoint, normality calculated, and the normality given of perchloric acid. Next is the estimation of ephedrine hydrochloride. How is the estimation of ephedrine hydrochloride done? We will be discussing that. First of all, ephedrine hydrochloride has the molecular formula C10H15NO, and with that hydrochloride is being associated. The molecular weight of the compound is 201.7. It is colorless crystal or white. It exists in a crystalline form. It is odorless and it is light sensitive. It is affected by light. How is the estimation of ephedrine hydrochloride done? We will be discussing the procedure. Now, the principle which is involved in the estimation of ephedrine hydrochloride is the chloride ions which are present in the ephedrine hydrochloride is a weak proton acceptor. The chloride ions, because of the negative charge on them, they are going to act as weak proton acceptor. This chloride ion is replaced by the acetate ion with the addition of mercuric acetate. On the addition of mercuric acetate, the chloride ions will be replaced by the acetate ions. Acetate ions have much higher tendency to accept the proton as compared to the chloride ions, and hence they can give us more accurate endpoint when titration against perchloric acid is done. Known amount of ephedrine HCl is titrated with perchloric acid by using glacial acetic acid as the solvent, and the endpoint is detected by using methyl orange as indicator. The reaction which takes place is as follows. Ephedrine here releases chloride ions. The chloride ions here are released. This is the main reaction which is happening here. Now the acetate ions from the mercuric acetate are going to replace the chloride ions. The chloride ions which have been produced by the ephedrine will be replaced by the acetate ions. So when we react mercuric acetate with chloride ions, we will be getting mercuric chloride with the formation of acetate ions. So these acetate ions now can be estimated very easily. So here we have estimated the strength of ephedrine indirectly by, first of all, dissociating chloride ions. The number of chloride ions are exactly equal to that of ephedrine present, and by the chloride ions have been replaced by the equal amount of by the equal amount of acetate ions. From the acetate, since the acetate ions are much stronger base, and hence they can give us more accurate endpoint. So hence this estimation will be done. How will be the estimation done? As ephedrine is going to release chloride ions, the number of chloride ions which we will be getting is equal to the ephedrine present with us. The chloride ions, the number of chloride ions then will be exactly same to that of the acetate ions which we will be getting. So indirectly, by estimation of acetate ions, we are finding out the number of chloride ions, and by that indirectly we are finding out the concentration, the strength, or we are performing the estimation of ephedrine. So the acetate ions will react with onium ion. Onium ion has been produced by the protonation of the glacial acetic acid by the perchloric acid. The onium ion will react with acetate ion to form acetic acid. So in this way, estimation of ephedrine hydrochloride is being done. Now we will study the preparation of perchloric acid, and we will be studying the standardization. First of all, as per the same protocol which has been mentioned earlier in the preparation and the standardization of perchloric acid, the same protocol will be followed while the preparation of 0.1 normal solution of perchloric acid and its standardization. Now, how will be the assay procedure done? Firstly, we will be weighing accurately about 0.17 gram of ephedrine hydrochloride. We will dissolve it in 10 ml of mercuric acetate solution. When we will dissolve 0.17 gram of ephedrine hydrochloride into mercuric acetate solution, ephedrine hydrochloride is going to release Cl negative ions, which we know will be replaced by the acetate ions from the mercuric acetate, and hence we will be getting acetate ions. Acetate ions, being a very strong base as compared to chloride ions, will react with the onium ions which we have produced by the protonation of glacial acetic acid by perchloric acid. Warming, after the addition of ephedrine hydrochloride and addition of mercuric acetate solution, we will warm the mixture gently so that homogeneous solution can be obtained. We will add 50 ml of acetone into it. Acetone here will not will just act as a solvent. It will not be interfering in the reaction. We will mix the solution thoroughly so that homogeneous solution is obtained. Then we will titrate it with 0.1 molar perchloric acid by using 1 ml of saturated solution of methyl orange in acetone as indicator. So the indicator which we will be using here is methyl orange. We will be adding 1 ml of the saturated solution of methyl orange which has been prepared in acetone, and we will be carrying out the titration till the red color is obtained. In the same way, we will also be performing the blank titration. How is the blank titration done? In the blank titration, we will not be adding ephedrine hydrochloride, and we will find out again the volume of perchloric acid which has been used. By performing the blank titration, we will come to know the volume which has been used, which has the volume of this perchloric acid which has been used by the solvents for their neutralization. So by subtracting the volume of that used by the ephedrine hydrochloride sample and to that of blank titration, we will be getting the exact value of the volume used for the estimation of ephedrine hydrochloride. Now, what is the equivalent factor? The equivalent factor here, 1 ml of 0.1 molar perchloric acid is equivalent to 0.02017 gram of ephedrine hydrochloride. So, by knowing the volume, by knowing the amount of perchloric acid which we have added, we will be coming to know how much ephedrine is present in our sample. So, that will come out to be, for example, if we use 20 ml, so we will just multiply 20 by the equivalent factor, that is 0.02017 gram, and we will be coming to know the amount of the ephedrine hydrochloride which is present in the sample, in the unknown sample. So hence the estimation of ephedrine hydrochloride can be done. Now, this is some more portion which includes the calculation. The percentage of ephedrine hydrochloride is equal to xml, that is the volume of titrant, volume of the perchloric acid which we are using, normality into 0.02017, that is the equivalent factor, into 100 divided by n normality which is given divided by the weight of the sample which we have taken. So by this estimation, can be done, and the percentage of the sample present can be analyzed. Now, next topic is buffer solutions. So the next topic is buffer solutions. We will discuss that now. Okay, so what are buffer solutions? Buffer solution is basically a mixture containing weak acid and the conjugate base of the weak acid. What is a buffer solution? It is a mixture which contains weak acid and the conjugate base of the weak acid, or we can say that it is a mixture which contains weak base and the conjugate acid of the weak base. So this is the buffer solution. It contains weak acid and the conjugate base of that weak acid, or we can say buffer solution is a water solvent based solution which contains a weak base and the conjugate acid of the weak base. Buffer solutions, why are they known as buffer solutions? They are those solutions which resist any change in the pH on the dilution of addition of small amounts of acid or alkali. When we add small amount of acid or alkali into the buffer solutions, they do not change their pH. They have the capacity to resist any change in the pH on the addition of small amounts of acid or alkali to them. So these are the buffer solutions. We will read it once. Buffer solutions are water solvent based solutions. They are prepared in water, which consist of a mixture containing a weak acid and the conjugate base of the weak acid, or a weak base and the conjugate acid of the weak base. They are buffer solutions. They resist change in the pH upon the dilution of on addition of small amounts of acid or alkali. If we add small amount of alkali or acid to it, they do not show any pH change. pH of the buffer solutions show minimal change, very less change upon the addition of very small quantity of strong acid or base. Therefore, they are used to keep the pH at the constant value. So why is the addition of buffer solution done? The buffer solutions are added to those solutions whose pH is to keep when we are to keep the pH of the solution constant. Then buffer solutions are used, because buffer solutions do not show any change, or they show very minimal change upon the addition of small quantity of strong acid or base. Now, the buffer solution is a solution which is able to maintain its hydrogen ion concentration. That is, it is able to maintain its pH with only minor changes in the dilution or addition of the small amount of either acid or base. It is the buffer solution is able to maintain its hydrogen ion concentration with the addition of small amount of either acid or a base. Buffer solutions are normally used in fermentations. They are used as food preservatives in drug delivery, electroplating printing, activity of enzymes, blood oxygen carrying capacity, need specific hydrogen ion concentrations. All these processes like fermentations, food preservations, drug delivery, electroplating, printing, the activity of enzymes, blood oxygen carrying capacity, they all need a specific pH concentration. So to maintain that, buffer solutions are used. Solutions of weak acid and its conjugate base, or weak base and its conjugation acid, they are buffer solutions. They help us to maintain the pH. Now the buffer solutions are of two types. The two types of buffer solutions are acidic and alkaline buffers. We will discuss them. What are acidic buffers? As the name suggests, these solutions are used to maintain acidic environments. Acidic buffers, they are used to maintain acidic environments. And how are they prepared? First of all, acidic buffers, they have acidic pH, and they are prepared by mixing weak acid and its salt with a strong base. So how is the acidic buffer prepared? Acidic buffer is prepared by mixing weak acid with its salt with a strong base. An aqueous solution of an equal concentration of acetic acid and sodium acetate, which has a pH of about 4.74, can is normally used as acidic buffer. Now the pH of the acidic buffer is generally below 7. These solutions, as I told you, contain weak acid and the salt of weak acid. The examples is the mixture of acetic acid and sodium acetate. Now acetic acid is a weak acid, and the salt of sodium acetic acid, we have prepared the salt of acetic acid with a strong base. Strong base can be sodium hydroxide. When sodium hydroxide reacts with acetic acid, it releases sodium acetate as salt. So hence the acidic buffer, it consists of a mixture of sodium acetate, which is the salt, of the weak acid, that is acetic acid, with a strong base, that is sodium hydroxide. Now next are alkaline buffers. As the name suggests, they will be used to maintain the basic conditions. Alkaline buffers are used to maintain the basic conditions. Basic buffers can be prepared by mixing weak base and also the salt and its salt with the strong acid. So how are the basic buffers prepared? They are prepared by mixing a weak base and its salt with strong acid. The aqueous solution of equal concentrations of ammonium hydroxide and ammonium chloride has pH of about 9.25. Ammonium hydroxide is a weak base. Its salt has been prepared by treating ammonium hydroxide with HCl solution, which is a very strong acid, and hence ammonium chloride is prepared. The pH of this alkaline buffer is about 9.25, which is above 7, which is to the alkaline side. The pH of these solutions is above 7. And the example, we have discussed the example of alkaline buffer solution, that is a mixture of ammonium hydroxide and ammonium chloride. The pH is towards the basic side, that is 9.25. Now, let us discuss the mechanism of buffering. We will be discussing the mechanism of buffer solutions. In the solutions, what happens, the salt as well as the weak acid, they ionize. So what happens in the, we will be considering here acidic buffer. Acidic buffer was prepared by mixing acetic acid and sodium acetate. So what is going to happen? Both acetic acid is going to release, it is going to act as acid, it will release H positive ions and it will form acetate ion. Similarly, the salt, sodium acetate, will dissociate itself into sodium ions and acetate ions. Now here we have acetic acid, we have acetate ion from both the equations. From equation number 1 as well as equation number 2, we are getting acetate ion. On what happens to the solution when we are going to add acid or base. So what is going to happen on addition of acid or a base? On the addition of acid, when we will be adding acid from outside, the released protons of the acid will be removed by the acetate ions to form an acetic acid. Now, if we are adding H positive acid, that is, we are adding acid from outside, we are adding protons from outside, what will happen? They will combine with the acetate ion. They are going to combine with the acetate ion to form acetic acid, and hence the protons will be consumed. The protons, the acid which we have added from outside, will be consumed by the acetate ions which were already present in the acidic buffer, and hence since we have used all the protons which we have added from outside, so there will be no change in the pH of the solution. So this is the mechanism which is working behind the buffer solutions. the protons, the acid which we have added from the from outside, are removed by the acetate ions to form acetic acid, and hence the pH, the number of proton ions will be the same, and hence the pH will also be the same. If we are doing the addition of a base, if we add a base in our buffer, what happens? The OH negative ions which will be released by the base which we have added from the outside, they will combine with the hydroxide ions which were already present in the buffer to form water. So what is going to happen? The hydroxide ions which we have added from outside, they will combined with the protons which were already dissociated, which were already released by the released by the acidic buffer to form water. So hence again the OH negative ions are no more left in the solution. They have been completely neutralized by the proton ions from the buffer, and hence the pH will again remain the same. How is the preparation of a buffer solution done? If the dissociation constant of the acid pKa and that of the base is known to us, the buffer solution can be prepared by controlling the salt acid or salt base ratio. The equation which here we will be applied here will be Anderson-Hasselbalch equation. We will discuss that equation. By knowing the dissociation constant, we can easily prepare the buffer solution. As discussed earlier, these solutions are prepared by mixing weak bases with their corresponding conjugate acids, or by mixing weak acids with their conjugate bases. An example of this method is an example of preparing buffer solution is by preparation of how can the phosphate buffer be prepared? It can be prepared by mixing HPO4 negative and H2PO4 negative. The pH maintained in the solution is approximately 7.4. Now we will be considering the preparation of acidic buffer. Consider an acid buffer solution containing a weak acid and its salt Ka, that is with a strong base. What happens here? Weak acid is going to ionize, and the salt is also going to ionize into K positive and E negative ions. We will be applying the Henderson-Hasselbalch equation. According to that, pH is equal to pKa minus log of salt, concentration of salt divided by that of acid. So which equation we will be applying here? That is pH is equal to pKa minus log concentration of the salt divided by the concentration of the acid. By using this concentration, we can easily find out the dissociation constant. So from here we will take pKa on the left hand side and we will shift the pH on the right hand side. From here we will be getting the acid dissociation constant, that is Ka is equal to the concentration of H positive ions and multiplied by the concentration of A negative ions divided by the concentration of the salt we have taken, that is Ha. Now we will be taking the negative log on both the sides. After taking the negative log on both the sides, we will get this equation. We will be getting this equation finally. And now the next one is the preparation of base buffer. Consider base buffer solution. As we know that base buffer solution consists of a weak base and its salt with the strong acid. So how can we calculate the POH value? The POH value can be calculated by the same equation, that is we can be calculating it by using the same Henderson-Hasselbalch equation. Firstly, we will find out the POH value by using that equation. It is similar to that which we have studied here. In place of pH, we will be having POH, and instead of pKa, we will be having pKb. So what is this equation? This equation will be modified to POH of a basic buffer is equal to pKb plus log concentration of the salt divided by the concentration of the acid. So from here we will calculate after this calculation we will be able to calculate the POH, and from the POH we can easily calculate the pH value. Now, what is the significance of using Henderson equation here? It will help us to Henderson equation can be used to calculate the pH of the buffer prepared from the mixture of salt and the weak acid or base. So we can find out the pH also if we know the concentration of the salt and that of weak acid or base. We can find out the pH, we can find out the pH of the buffer. Secondly, we can calculate the pKa value. If we know the pH and we know the concentration of the salt and base, we can calculate the pKa value also. Third point is we can prepare by using this equation, we can prepare the buffer solution of the needed pH. What is the limitation of this equation? What the limitation of Henderson-Hasselbalch equation is that it cannot be used for strong acids and strong bases. It can be only used for weak acids and weak bases. It cannot be used for strong acids and strong bases. Next important term is buffering capacity. What is buffering capacity? Is the number of millimoles of acid or base that is needed to a liter of a buffer solution to change the pH by 1 unit. So how much millimoles of acid or base must be added to 1 liter of the buffer solution so that the change in the pH can be made by 1 unit. This is known as buffer capacity of the buffer. So this is basically the millimoles of the acid or base which we have to add to 1 liter of the buffer solution to change the pH by 1 unit. So this is basically the formula. So buffer capacity is equal to the millimoles of acid. It is equal to millimoles of acid or base which have been added to 1 liter of the buffer solution to produce pH change by 1 unit. So there now we will be discussing few problems of buffer solutions. So the first problem is what is the ratio of base to acid when pH is equal to pKa in the buffer solution. So this is the first part. So this question can be solved by using the previous equation. Henderson-Hasselbalch equation. According to this equation, pH is equal to pKa plus log concentration of salt over concentration of acid. So when will be the value of pH is equal to pKa plus 1, it means that the term which is in the log side should be equal to 1. So when is log equal to 1? That is when log, the value of log will the inner portion has to be 10. The value of the concentration of the salt to that of the concentration of acid must be equal to 10. Only then we can get 1 in the equation. So first of all, before solving this equation, when the concentration of salt and acid is equal, in that case, the ratio of the base to acid is 1. So the log 1, as we know, log 1 is equal to 0. When is log base over acid equal to 1? When the ratio of the base to acid will be equal to 10. The ratio will be 10 is to 1. What is the pH of the buffered solution of 0.5 molar ammonia and 0.5 molar ammonium chloride when enough hydrochloride acid corresponding to make 0.1 molar HCl? The Kb value is given. From the pKb value, we can calculate the pKa value, that is pKa value plus pKb value is equal to 14. So we know the value of pKb. From that we will be calculating the value of pKa, that is 14 minus pKb, and then we know that 0.15 molar H positive ion reacts with 0.515 molar ammonia to form the salt, to form ammonium salt. So the remaining ammonium ion, that is 0.65 molar, and the 0.35 molar remaining ammonia base, we will be using Henderson-Hasselbalch equation on that. As we know the value of pKa which we have calculated above, that is 9.25. We know the concentration of ammonium ion and ammonia. We will apply this equation and we will calculate the value. We will hence be able to calculate the value of pH. We will be knowing the value of pH from this equation. Now problem number 2 is how many moles of sodium acetate and acetic acid must be used to prepare 1 liter of 0.1 mole per liter buffer with pH 5. So again we will be using the equation which we have used previously. pH is equal to pKa plus log concentration of the salt and the concentration of the iron divided by the concentration of the salt. pH here is 5. It is given to us. The pH is 5. pKa value of acetic acid, we know that it is 4.74. From here, we can calculate the concentration of the ionic form and the salt form in the undissociated form. We will be taking log on the other side, which will change into anti-log, and we will be calculating the value which comes out to be 1.82. From here, we can calculate the number of dissociated A negative ions by multiplying 1.82 with the concentration of the acid. Also, it is given to us that the concentration of A negative ions and the concentration of the acid is equal to 0.1 mole per liter. So we will put the value of A negative, the concentration of A negative ions here. We will put that in this equation, and by the substitution, we will come to know the value of acid, that is Ha. By knowing the value of Ha, we can substitute that in this equation. The concentration of A negative ions is equal to 1.82 into concentration of the acid. So we can, by the substitution, we can come to know the value, and hence we can calculate the number of moles. From A negative, we can calculate the number of sodium acetate ions which has been used to prepare the solution, 1 liter solution of the buffer solution. So next topic is pH maintenance. In order to understand how buffer solutions maintain a constant pH, let us consider a few more examples. Let us consider the example of buffer solution containing sodium acetate and acetic acid. For in this example, it can be noted that sodium acetate almost completely undergoes ionization. Whereas acetic acid is only weakly ionized. So what is going to happen? The salt as well as the base, they are going to dissociate. Acid is going to dissociate into H positive ions and acetate ion, whereas the salt, that is the sodium acetate, is going to dissociate into sodium ions and acetate ions. When strong acids are used, when we will be using strong acids, when we will be adding acid from outside, the H positive ions will combine with the acetate ions to form weakly unionized acetic acid, and hence there will be no pH change on addition of H positive ions. When strongly alkaline substances, those substances which can release OH negative ions, they are the strongly alkaline substances, are introduced to the buffer solution. The hydroxide ions, they react with the acids, they react with the H positive ion, or they react with the acetic acid which we have produced. They will react with them to form salt plus water, and hence our OH negative ions which we have added from outside has been consumed. Since no OH negative ions are present now, the pH will remain same. There will be no effect on the pH, and hence the pH of the solution will remain the same. So now we will be discussing few uses of buffer solution. There exist a few alternative names that can be used to refer buffer solutions. Buffer solutions are also known as pH buffers or hydrogen ion buffers. An example of the use of buffers in the pH regulation is the use of bicarbonate acid and carbonic acid buffer in order to regulate the pH of the animal blood. So in order to maintain the pH of the animal blood, we use bicarbonate and carbonic acid. Carbonic acid is a very weak acid, so it can be used to prepare acidic buffers. Carbonic acid, along with its salt with strong base, can be used to prepare acidic buffer. Buffer solutions are also used to maintain an optimum pH for the enzyme activity in many organisms. So pH solutions are also used to maintain the optimum pH for enzyme activity in many organisms. The absence of these buffers may lead to the slowing of enzyme activity. If the pH changes, the enzyme may not work, because as we know that enzymes are very specific to pH, and hence the use of buffer solutions is very important. Without the buffer solutions, the pH may change and there might be slowing of the enzyme action, and hence the loss in the enzyme properties, or it may also result in denaturation of the enzymes. So this denaturation process can even permanently deactivate the catalytic action of the enzymes. Hence the buffer solutions are really important. Now we will be discussing few questions based on this important questions. The first question is, discuss the non-aqueous titrations of weak bases with perchloric acids. We have discussed the reaction of we have discussed the non-aqueous titration of weak bases with perchloric acids by considering pyridine as one of the examples. In that case, we have studied that firstly we have prepared the solution of perchloric acid in acetic acid. Since perchloric acid is a very strong acid as compared to acetic acid, so perchloric acid is going to protonate the acetic acid and it will form onium. Now the onium which has been produced will act as an acid, and then we have also prepared the solution of the weak base by taking an example of pyridine in acetic acid. By dissolving pyridine in acetic acid, the lone pair which was present on on pyridine was protonated, and after that, after preparing the solutions of both base and perchloric acid in acetic acid, the titration was done. And the next question is, discuss the non-aqueous titrations of weak lithium, weak acid with lithium methoxide. So the next was the titration of weak acid with a weak base. In this, we have also studied how the solutions were prepared. In this case, the solutions were prepared in dimethyl formamide, and then we have studied the titration of the weak acid with lithium methoxide. uh, for earlier, first we have prepared the solutions followed by the standardization. We have studied that the standardization of solutions is very important, and after the standardization of solutions we have performed the titration of weak acid with the lithium methoxide. What are next question is, what are the applications of non-aqueous titrations? We have discussed the applications of non-aqueous titrations. In the applications of non-aqueous titrations, we have studied that the water cannot be used, aqueous titrations can never be always be used, because almost most of the drugs are weak acid or weak base. If we perform the titrations of weak acid or weak base in water, then water will compete with the acid or the weak base which we want to whose a quantitative analysis we want to make. So water should be omitted in those reactions, and hence non-aqueous titrations are used. The next